Assalamualaikum. I am Faiqa Sajid from Department of Physiology, Government College University, Faisalabad. Firstly, I'm highly obliged to the Department of Zoology for giving me the opportunity to present in the 4th International Conference on Applied Zoology 2021. The topic of my presentation is the role of methanolic extract of Indian ginseng Vidania somnifera in promoting functional regain in mouse model of peripheral nerve injury. Let's start with the introduction. As we know, the nervous system is our command body system, and the central nervous system controls and coordinates all body function with the help of brain and spinal cord, whereas the peripheral nervous system connects the central nervous system through the nerve fibers. Any damage to the peripheral nerves due to vehicle accident, laceration, or any other reason may result in the devastating neurological functional loss with unpredictable functional recovery despite optimal medical management. When an injury occurs, the nerve injury is followed by two sides of changes. First one is the proximal side of nerve injury, which includes degenerative changes and regenerative changes. Whereas at the distal side of the nerve injury, only bleeding degeneration occurs, which is necessary to promote the nerve regeneration. After an injury, the axon gets demyelinated and loses its power of conduction. Peripheral nerve injury usually undergoes muscular atrophy due to slow rate of regeneration, which is 1 to 3 millimeter per day. Injured axon usually required 3 months to reinnervate the target organs. Meanwhile, the target organs undergo muscular atrophy, which results in neurological function loss. Available treatments includes surgical intervention and non-surgical intervention, whereas non-surgical interventions are some medications, electrical nerve stimulation, and phytochemicals. And surgical interventions are direct nerve repair, nerve grafting, nerve transfer, nerve conduits, fibrin glue, and cell-based therapy. But both repairs from surgical interventions and non-surgical interventions are not capable of fully nerve regeneration. Moreover, they are time-consuming and expensive. Therefore, they cannot fully support the nerve regeneration after an injury. In order to improve the motor function recovery, we need to reduce the time required for the peripheral nerve regeneration. Phytochemicals, plants are full of bioactive compounds known as phytochemicals, and they have several pharmacological properties like antioxidative, anti-inflammatory, analgesic, antimicrobial, and neuroprotective. Along with these effects and having no side effects, there might be the possibility that they can accelerate the functional recovery after a nerve injury. The aim and objective of this study was to assess that can Vidania somnifera can accelerate the functional recovery for injured sciatic nerve. Vidania somnifera is a well-known plant for its various pharmacological activities. It is also known as poison gooseberry, skunk, and Indian ginseng, and it is reported to have effective antioxidative, anti-inflammatory, antipyretic, and many others. It is also effective in the Parkinson's disease and various other neurological disorders. We use the mouse model in which sciatic nerve was induced at mid-thigh region. After anesthesia, the compression was given to the sciatic nerve with the help of forceps. It mimics the human nerve injuries after road accidents. After injury, 100 milligram methanolic extract of Vidania somnifera root powder per kg body weight were given to the treatment group through the gouging until the end of the experiment. For analysis, different behavioral and biochemical parameters were performed. Behavioral analysis include food intake analysis, weight analysis, working track analysis, grip strength, and heart rate analysis. Biochemical analysis include total oxidative stress, total antioxidant capacity, and blood glucose measurement. We found some interesting results. There was observed that food intake was normal in all groups, confirming proper treatment, and there was no change in body weight, which means that treatment has no effect on the body weight. In the case of grip strength, it was observed that earlier force gain was observed in treated groups, and similarly, SFI also shows accelerated motor function recovery, which means the treatment groups was effective in motor function regain. For hot plate analysis, there was also reduced latency period, which means the sensory function restoration was gained in treatment groups. For blood glucose level measurement, it was observed that blood glucose level was decreased in treated group, which means this plant has glucose lowering effect. Lastly, the total antioxidant capacity and oxidative status was measured, which the observation shows that treated groups reduced oxidant level and increased the antioxidant capacity. Based on these results, we may say that Vidania of somnifera roots offer meliorating effects on the function recovery after an injury. But we need to identify the biotic molecules and characterization and further identification of signaling pathways. Thank you for the attention.